Thank you for that introduction. It's a real honor to have the opportunity to give this talk. I'm sorry I cannot be there to connect with everyone in person, but that's something to look forward to for the next meeting. And so I'm going to be talking about transcranial direct current stimulation and its application to neurological and psychiatric disorders, but specifically thinking about how tDCS might help the brain heal after injury. And in that sense, I'm going to be talking about plasticity and essentially, the ability to teach the brain how to do new tricks. These are some of my relevant disclosures. And for those who are interested, these slides are already online on my Twitter, as well as a link to all the papers that I will be discussing as well. There's different forms of neuromodulation or brain stimulation technologies. They have different names. Usually the name of the technology tells you how it gets energy into the body or where it tries to deliver the energy. So for example, deep brain stimulation will involve implanting electrodes deep in the brain and electrically stimulating those targets. You have technology like transcranial magnetic stimulation, where a coil is placed over the head. It is transcranial, it is non-invasive. And then you have technologies like transcranial electrical stimulation that place electrodes on the scalp. And one way to classify these is there are certain technologies that require surgery. There's an implant, deep brain stimulation, spinal cord stimulation. There's certain technologies that would typically require an inpatient or at-center treatment, such as transcranial magnetic stimulation or ECT, electroconvulsive therapy. And this is due for technical or safety reasons. And then there's this last bit of technologies, these transcranial electrical stimulation technologies, including transcranial direct current stimulation, which to use a popular term, they're wearable. They're battery powered. You can theoretically walk around your house while you're wearing them. And so I'm going to be talking about one type of wearable neuromodulation technology, and that's transcranial direct current stimulation. So transcranial direct current stimulation is unique in the world of neuromodulation in that almost all other technologies use pulses, like puck, puck. So you'll talk about, the frequency of transcranial magnetic stimulation or the frequency of DBS, or talk about the frequency of the pulses. Transcranial direct current stimulation has no frequency, or it has a zero frequency because it is a continuous wave. Other than initial ramp on and a ramp off, it's on the whole time. So it is analogous to a 9-volt battery. And that's what's being applied across the head, a direct current, transcranial, because it's non-invasive, it's scalp electrode, direct current, because of the waveform, and stimulation, because the goal is to produce brain stimulation. And it is an extensively investigated technique across indications, both in healthy individuals to change cognition and behavior in healthy individuals. Often college students are the ones who are recruited. And then a wide range of neurological and psychiatric disorders, including neurorehabilitation. So this is what it actually looks like. This is on my head. You can see that there are these two pink electrodes positioned on my head, and they're being held there by a plastic headgear. And if you look closely, you can see that there are some wires running down the side of my head, a red wire and a black wire. And those wires are running to connect, each wire is running to one of those pads. So the pads are called electrodes, and current flows from one pad to the other pad, so across my head. One pad is connected to the positive terminal of the stimulator, or call it the 9-volt battery or the current source. We call that the anode, the positive electrode. The other is connected to the negative terminal of the battery. We call that the cathode. So we have anode and cathode, and current flows from anode to the cathode, and what's in the middle but my head and my brain. And so in that way, we deliver current across the brain. It's a very low level of current. So, we can use a 9-volt battery analogy. These devices really are powered by batteries, 9-volt batteries. So we express the current in terms of milliamps. It's a current control device. So it's 2 milliamps of stimulation would be typical, 1 sometimes. And the stimulation lasts for about 20 minutes. So a single session would be 20 minutes. And then those sessions might be repeated. It might be daily, it might be just one, it might go on and on. Now, you'll often hear the terms anodal or cathodal transcranial direct current stimulation. I want to emphasize there's always an anode and there's always a cathode. So if I say anodal, what I mean is that the brain target that I'm interested in stimulating is near the anode. And if I say cathodal, what I mean is that the brain target that I want to stimulate is near the cathode, but they're always both there, and we're always doing both the nodal and cathodal stimulation. I mentioned that it's a widely investigated technique, including on college students. And the reason we're allowed to do it on college students routinely is it's considered to be very safe, and it's also very tolerated. The main thing people feel is like prickling. I'm like an itcher. I get an itching sensation. And when you remove the electrodes, you see skin redness, you see erythema right under the under both electrodes. And that all this, the redness resolves after 5 minutes, and the tingling resolves as soon as the stimulation is turned off. It's very low intensity. And so other than that, there's no overt brain response. It's not like transcranial magnetic stimulation, for example, where you get a finger twitch or other techniques. It is a very nuanced and mild intervention. And I'll be talking about how that's really considered a feature of it. I really want to emphasize this notion of the anode and the cathode. The anode is a positive electrode where current flows into the brain. The cathode is the negative electrode where current flows out of the brain. So there's current flowing both under the anode and under the cathode. The only difference is the directionality. And this directionality is considered to be very important in how direct current stimulation affects the brain, and also why there's a difference between anode and cathode. Under the anode, the soma of cortical pyramidal neurons is assumed to be or understood to be depolarized. So this is a nodal stimulation under the anode, producing a depolarization of the soma. And decades of work, literally decades of work, have shown this to increase excitability, increase firing rate, increase propensity for plasticity. And so we'll use a colloquial term and just say, so it's a boosting effect. And I'll talk more about that. Now, under the cathode, there is still current, the same amount, but in the opposite direction. Because it's the opposite direction, the soma of these pyramidal neurons is hyperpolarized. So cathodal tDCS, meaning under the cathode, we have soma hyperpolarization. And there's been a lot of work to show that this has a dampening effect. So hence, a nodal excitatory and cathodal inhibitory is a simplistic but starting point for how we think about tDCS and how we rationalize tDCS for different indications. For example, let's say there's a part of the brain that has been injured due to stroke or for some other reason. It may be a strategy to put the anode electrode over the part of the brain that has been injured with the goal of boosting its function, perhaps promoting plasticity or promoting plasticity in the perilesional region around it, or otherwise increasing the capacity of the brain around this region. So again, that's a simplistic approach, but it's one that's actually gotten us very far in tDCS. So here's one example of a paper from Julius Fredrickson's group, published now over a decade ago, where transcranial direct current stimulation, tDCS, was investigated as a treatment for aphasia. And there in the conclusions, you can see a nodal tDCS over the left frontal cortex. So there is an anode that is in B. There is a cathode that is in C. In this case, the cathode is on the arm, but the anode is placed over the brain target. And so we have a nodal form of stimulation. And this particular trial produced some very encouraging results that led to a lot of subsequent work on the topic. And so I want to unpack this picture here because I think it raises a lot of very interesting questions about transcranial direct current stimulation. Like, where should you put the anode electrode? And where should you put the cathode electrode? Because you have to put both of them somewhere. You can't just have one. It's not possible. You have to have an anode and a cathode. tDCS, when used for many indications, including for aphasia treatment or generally for neuro-rehabilitation, is paired with a task. So what task should it be paired with? Should it be a special task that's somehow well matched to tDCS? Should it be the tasks that you are already very familiar with using? And then related to that, this is point number D, what intensity should it be used? 2 milliamp, 1 milliamp? Should it go on for 20 minutes? Should it be applied before the training, during the training, maybe after if you're interested in some kind of consolidation? So I think this picture nicely wraps up what I think are really essential questions in how we apply tDCS. So not just how does tDCS work, but also how do you employ it optimally, depending on your indication. And I'm happy to say I will not be addressing these questions. Fortunately, there's a wonderful session coming up with really the absolute leaders in the clinical trial space. And so I will defer the conclusive answer to all these questions to them. So you can save their questions on clinical deployment for that. But I do want to generally speak about these questions from a mechanistic and from a technology perspective. And I want to start with this question of where you should place electrodes, because this is something that we think a lot about and I think is an essential decision in tDCS. And actually relates to the start of at least my collaboration with researchers who deal with treatment after stroke, in particular aphasia. And this is a paper that we published using computational models to try to figure out how to place electrodes in the case of stroke and in the case of the patients with aphasia. But before I talk about that, this was almost a decade ago. I need to go back a full decade to describe the actual modeling approach. So this modeling approach that we developed in a general context for any kind of head, then is then applied for stroke application. So briefly, this was what we came up with. We came up with a modeling pipeline that starts with an MRI, either from a healthy individual or it could be an atypical MRI. And we essentially segment the tissue into different masks, such as skin, skull, CSF. And the masks that we're considering are tissue types with very different resistance. So for example, cerebral spinal fluid is much more conductive than skull. So it's very important for us in our models to separate them out. Once we've done that, we place the electrodes on the model as if you would place it on the person. And then we predict how current flows through the head. So based on the individual anatomy, and based on the given electrode placement, we ask the question, where in the brain does the current go? Like, does it go just under the electrodes? Does it flow somewhere in the middle? Does it flow all over? That's the question we wanted to answer. And so let me show you one example of that. This is with the M1 SO montage. M1 is for motor. One of the electrodes is over the motor cortex. SO stands for supraorbital. It's over the eye. And so we simulated the M1 SO montage. This is on a healthy head model. And there you can see the electrodes. They're 5x7 pads. And we simulate the current as it flows through all the structures, through the whole brain. But the results I'm going to be showing you are on the surface of the brain itself. That's the target that we're interested in. And where current flows is going to be shown in false colors. So the hotter colors mean there's more current flowing there, while the darker colors mean there's no current flowing there. And that is a representation of electric field or how much electrical energy is in each spot. So this is a result of one of these simulations. This is the M1 SO montage. And briefly, what you see is a lot of red with a few little yellow hot spots. And those yellow hot spots are not necessarily even under the electrodes. They're somehow in between. They're sort of clustered and on these gyri crowns. And so this model predicted that the M1 SO montage, and in general, tDCS using large electrodes produced current flow through much of the brain. And you can see through the cross sections that current also runs deep, very deep. It doesn't dilute with distance. And so the prediction was that tDCS with large pads, while you can guide where the current would go, you're stimulating a large trunk of the brain. And this was controversial. We also considered, instead of using large pads, using smaller electrodes and deploying them in different configurations. So these small electrodes were called high definition electrodes or HD electrodes. And we can deploy them in configurations like this. This is the 4x1 configuration, where you can imagine that the center electrode is the positive, and then there are 4 negative returns that are collecting a current. And once again, we, in a computer simulation, predicted where the current flow would go, and I'm showing you that result just on the surface of the brain. And the model predicted that when you do 4x1 stimulation, you can in fact be very focal. And you can in fact drive current to specific regions of the brain. And you can actually move this 4x1 ring anywhere you want on the cortex in order to stimulate a particular region. So this was all a computer prediction. And correctly, we had a lot of pushback from the brain stimulation community, from the neuromodulation community. And this is, a decade ago, this is where my talk would stop, and then the questions would come. First of all, the suggestion that conventional tDCS with large pads was not focal was inconsistent with how tDCS was being rationalized until then. It was also inconsistent with the finding that when you move these pads around, you can produce very different effects. However, having location-specific effects is not the same thing necessarily as being targeted. But there's a lot of pushback against the notion that conventional tDCS was not focal. At the same time, there was also a lot of pushback over the notion that you could be focal and non-invasive. So I mentioned technologies where holes are drilled in the skull in order to deliver the current focally. And so the notion that the skull, at the same time, there was this notion that the skull somehow made it impossible to be both focal and non-invasive. So on both fronts, there was a lot of very valid criticism from the community, which we and other people did overcome. Here's one of the experiments, I think one of the key experiments that help overcome that concern, and I think it's worth outlining. These were 3 subjects that were tested. They were actually the investigators. The top one is that's me, that's my MRI, and those are the masks that have been segmented in this modeling flow. And you can see that there's 2 other individuals with different head shapes, and all their tissues are very different. We did 4x1 stimulation on all of us, but we didn't do tDCS with 2 milliamp. Instead, we applied pulses at 1,000 milliamps. Very high intensity pulses for very short periods of time. When you do that, you produce a motor twitch. So when you increase the intensity that much, and when you pulse it, each pulse will produce a motor twitch. So it's a very obvious readout that the current is getting into the brain. And we produce these motor twitches on all 3 subjects using the 4x1 montage. We took it a step further, and I mentioned that you can move that 4x1 back and forward. So what you're seeing there in the column is a model that predicts when you put the 4x1 over the motor strip, the current gets to the motor strip. But when you move it anterior or posterior by just a few centimeters, the current entering the brain is no longer, it's still entering the brain, but it's not in the motor strip anymore. And so the model predicted that you would no longer get a motor twitch. And so what we did was, in the experiment, which is where it's seen in the other column, is that we started by producing a motor twitch, where it says TES, and what you're seeing there is the motor evoked potential that indicates that the finger is twitching. And then we would move these rings forward and back, and the twitch, sure enough, would go away. By the way, we also did this at the same time with TMS. So the TMS coil was also being moved back and forth. And when either one was moved anterior or posterior, the response went away with roughly the same resolution. But there's one more thing that was really important here. It wasn't just that we showed that we could deliver the current focally through the cortex. We also found that there was very large differences in how much current we needed across subjects. And there's different ways to show that. One way that we found that is that on me, with my big head, we needed to crank the dial up all the way. I needed 1,000 volts to produce a twitch. But the other subject, subjects which had less of a big head, we needed much less stimulus to produce the same twitch, okay? And another way to do that same experiment is to give everyone the same intensity, and 600 volts did not produce a twitch on me because I needed 1,000 volts. Well, it was more than enough to produce a twitch in the other subjects. What that was telling us was that different subjects with different heads responded differently to stimulation. And more specifically, me, because of my big head, I needed more juice at the scalp to get enough current into my brain to produce a response. So the current path into the head depends not just on the montage, where we put the electrodes, but on the head itself. And it varies a lot across individuals. Different people have different shaped heads, and so the amount of current that reaches the brain is going to be very different. And so then the question comes up, let me go back here. So then the question comes up, well, if it varies so much on anatomically typical heads, how much would current delivery to the brain vary for anatomically atypical? And this brings us to the paper I discussed earlier, where we worked with Julius Fredrickson and his team, and we developed the first realistic model of how transcranial stimulation passes current through the brain when there's an injury. And this was this particular injury on this individual who did struggle with aphasia. And what we found was that the lesion mattered a lot. A large portion of the brain, which previously had gray and white matter, had now been replaced with cerebral spinal fluid. The brain is much more resistive than the cerebral spinal fluid. Cerebral spinal fluid is like salt water, as conductive as it gets. And so you replaced something that's very relatively resistive with something that's extremely conductive. And as a result, the whole current pattern through the brain changed. Specifically, this large sac of fluid was now really sucking the current in and changing how it was distributed everywhere. And all those details and the methodological details are in this paper as well. But it became very clear, even with this single case model, that the current flow pattern through the head of individuals with atypical anatomy, injuries in particular, would be altered. This is an idea that we kept building up on. I don't expect you to be able to see this table, but in this particular paper, what we did is we scanned across all the trials up to that point that had tried tDCS for aphasia treatment. Now, these trials had tried different montages that also used different tasks, that selected different types of patients. There were different inclusion criteria, and there was a mix of results. And we asked the question, if we ran a computer simulation of the different montages used, montage A, montage B, montage C, could we then use that insight to try to make predictions about which montage was best, or which montage was best for which indication? This work continued. This is even up until this year. This was a paper with primary patients with primary progressive aphasia. So you're having sort of a slow or a cumulative loss of gray and white matter. So you have now more CSF between the brain and the skull. CSF is a highly conductive barrier. And so the question is, well, essentially these people's brains are now further away from the electrode, they're further away from the skull, and there's this sac of highly conductive fluid around it, particularly over the region you want to stimulate. Will current flow be different in these individuals? And so this paper unpacked that in tremendous detail, looking at different variants of PPA. And again, I'll refer you to the reference for all the detail that was done there. So a decade of work. And that decade of work conclusively showed that if you have an atypical brain anatomy, if it's lesions, if it's atrophy, other forms of energy, the way current will be delivered to these individuals will be different. And so then that raised the question immediately, should tDCS be personalized to the patient? Should tDCS be personalized to the brain anatomy? Don't give everyone the same tDCS. Give people tDCS that has been adjusted given their anatomy. And as I mentioned, this work had gone on now for over a decade, including the development of the computational models themselves. So there's a lot of tool development over this decade as well by self and by other groups. And so I think it's very appropriate here speaking at this meeting that I can announce sort of the conclusive answer to this question based on this full decade of work. And that answer is a absolute definite maybe with a sometimes and a sometimes not. So we don't know the answer to this. And so let me talk about that. The tools are in place. The models work. The models are not broken. It is absolutely possible to take any patient MRI and take any electrode montage and model it. And there is a recipe for how to get to an optimal, in quotes, electrode placement. This is what that recipe would look like. You load up the subject MRI, you select the target region that you're interested in. Maybe it's a perilesional region, maybe it is a region on the other hemisphere that maybe for some reason you want to inhibit. We'll hear more about that, I think, from the other talks. You need to dial in the orientation of the structure. It turns out that matters as well, and that can be actually very hard to sometimes tell with injury. You've got to tell it whether you have a 2-channel stimulator or a 5-channel stimulator, and whether you want to stick to 2 milliamp or whether you're comfortable going up to higher currents and so on. And there's other information you need to give it, like whether you just really want to be focal and not stimulate the rest of the brain, or you want to get that target with as much intensity as possible and you don't really care what happens afterward. When you do that, you run the model and you will always get an answer. That's what we and others have done. And what these models tell you, so in this case, the constraint, by the way, was 4 electrodes. So that's where you're seeing 4 electrodes, 2 anodes and 2 cathodes. And these are 8 sample individuals. X marks the spot of where the target was. And in each case, the model predicted the optimal place, the position to place the electrodes. What's obvious here, without going into a lot of details, is that each individual has a different prescription. For each individual, the model is telling us that we should place the electrode somewhere else in order to achieve an optimal result. So from the technical modeling perspective, it's a very closed story, right? The optimal electrode placement is not the same for everyone. But this is not quite the full story, right? It's not the situation. There are some groups that have started to do individual modeling and individual adjustment, but it's certainly not the trend as a whole, and that's correct, because there are pieces of the puzzle that are still missing that suggest this. So again, I want to emphasize the technology is there. I don't think there's any question marks about how we should do these models. The math, there's different constraints of how you would optimize, but all that's there, the technology is there. The majority of trials to date, many of them encouraging, did not do this. The majority of trials to date used ad hoc approaches, and they used large electrodes. And by ad hoc, I mean either they were all placed in the same position on each subject, or the research team somehow used either imaging or either functional or anatomical imaging to identify an individual target and just place the electrode just generally over it. So just placing it over it, but without the benefits of models. And again, these results have been encouraging. And so the question is there any evidence that doing it differently would produce different results? And there's other questions as well that get into this. What is the target, right? What is the mechanism of therapy? To the sense that these questions remain generally open in aphasia treatment, then they also remain open in using tDCS to enhance tDCS as an aphasia treatment. And so and I'll talk more about this. So there's many underlying assumptions that are caked into how we even want to optimize tDCS, what our targets are. And in addition to electrode placement, there's all these other apparently evidently very critical factors to determine outcomes, such as what tasks we match them with and how we time tDCS relative to the task. And so all of these issues come together to create a much more mixed picture than just suggested by a model where you click a target and it returns the optimal electrode montage. And so it goes back to this bigger picture that we talked before from Julius Fredrickson's early study. And these are sort of this quad of questions that we want to address. And up to now, I've really been talking only about electrode placement, excitatory electrode placement, at the same time inhibitory electrode placement. But these other questions I want to talk about now are equally important to the story. And they involve, what does a person do while they're getting tDCS? And how should the tDCS be timed relative to that training, right? If the training takes an hour and we're only applying tDCS for 20 minutes, how do we square that circle? So I want to talk a bit about that issue. From a mechanistic perspective, tDCS is very different than most other forms of neuromodulation we're used to. And I've already hinted as to why. Most forms of neuromodulation use high intensity pulses. Those pulses get delivered into the brain one way or another, and they trigger they trigger firing in a population of neurons that are exposed to the current flow. So current flow is delivered to the motor cortex, and then neurons fire, and they fire with every pulse, right? So that's where the pulsing comes in. There's a high intensity pulse that makes them fire. If the pulses are coming in at 10 hertz, 10 times a second, we produce 10 blasts of firing every second. And so these approaches, which I'm going to call now super threshold approaches, because their intensity is above the threshold to make things fire, pace neurons. Because of that, any effects that we see down the line, therapeutic outcomes, changes in cognition, changes in behavior, are secondary to the pacing. And by the way, this is also why these techniques tend to produce very overt outcomes like twitching. So TES, which is the 1,000-volt form of stimulation, TMS, ECT, these all produce very overt responses because it's so high intensity. And so neuromodulation is always secondary to it. Now I want to talk about tDCS, and I'm also grouping with that transcranial alternating current stimulation, transcranial random noise stimulation. These are sort of in a family of low intensity stimulation, very low current applied to the scalp. When this current reaches the brain, a fraction of it reaches the brain, it is not enough to make the neurons fire. So whatever brain region is now being engulfed by the current, the colors I showed you previously, it's not enough to make them fire. Again, tDCS is about 2 milliamp. ECT is 1,000 milliamps. We're nowhere near enough in stimulation to make neurons fire, and so they don't. Now, does that mean there's no effect? And the answer is obviously not, we don't think so, but there is no de novo firing. If you are not firing, tDCS will not make you fire. However, the brain is not quiet. The brain is always active, right? Or your brain is active right now. There's always ongoing firing, ongoing plasticity, ongoing homeostatic plasticity. And so when we think about how low intensity stimulation works, we need to think about it in terms of ongoing activity, as in you've asked someone to read something, and that is currently activating broad circuits across their brain. And you ask them to practice reading. And the reason you're asking them to practice reading is you're hoping that you'll produce some sort of plasticity and some sort of lasting benefit in their inability to read. So there's always activity going on in the brain, whether we're not doing anything or whether we're practicing a task. And so the notion is that in this case, the current will could even envelop the whole brain, but only certain neurons will get the benefit of the current. Only certain neurons will be modulated by the stimulation. Only the ones that are already active, that are already near threshold, crossing threshold, only those circuits will get the benefit of tDCS. And so this is activity dependent, meaning only the active neurons will fire. And hence we come up with this term called functional targeting. Everything I've shown you before was anatomical targeting. We kept focusing on driving the current to specific regions. Now we're talking about functional targeting, where the circuits that will be influenced will be the ones that are already active. So that was a nice cartoon. Do we have evidence for this? A lot. I'm going to be showing you data from brain slices. These are rodent brain slices. Some of it will be hippocampal slices. Some of it will be cortical slices. However, the story that has come out of this work, which goes back 2 decades, both from my lab and many other labs, is very consistent. And it shows that tDCS is a powerful modulator of ongoing activity, a powerful modulator of ongoing plasticity, but extremely ineffective in generating activity in quiet neurons. Okay, and in this slice, we can actually generate both the anodal and the cathodal variation of stimulation. There's the slice, you can see it, that pancake. There's actually 2 of them. There's a lot of small glass microelectrodes that are recording activity. We can do that with a high degree of precision that would be very difficult to do in a whole animal. And we can apply direct current stimulation by passing current between those 2 wires that you see on your screen. We can pass it one way and we can pass it the other way. And one way corresponds to the anodal variant of tDCS, and one way corresponds to the cathodal variant of tDCS. We don't call it, there's no transcranial because the skull isn't there anymore, but we still got the direct current stimulation component of it. And so I'm going to just walk you through, I think, one really key and illustrative experiment. This experiment has 2 pieces to it. I'm going to talk about the 2 pieces. The first one is we apply theta burst stimulation to produce long-term potentiation without direct current stimulation, just theta burst stimulation. This is a very intense pulse stimulation that's applied with a small electrode. It's like a and you basically blast the tissue. What this blast does is it produces plasticity. The way we measure that plasticity is by looking at a excitatory postsynaptic potential or a field excitatory postsynaptic potential. It is a blip that measures the strength of connection between 2 populations of neurons, a synaptic connection. So we have a baseline strength in that synaptic connection, a baseline EPSP. Then we blast it with this theta burst stimulation. And then again, we probe the strength of that connection, and we find that after blasting it with theta burst stimulation, there is an increase in the strength of that connection, a strength in the size of the EPSP. And so theta burst stimulation is a very robust way to force synaptic plasticity to begin and to produce long-term potentiation in animal models. Now we can combine this theta burst plasticity induction with direct current stimulation, either the anodal or the cathodal variant. And these are the results that we see. So there's our neuron, where there's an electrode we're using to produce the theta burst stimulation and also to probe the strength of the synaptic connection. There's a recording electrode, there's 2 field wires. And you can see there in the top, the pattern of theta burst stimulation. It's like a pattern of pulses, like a that's designed to produce it. And we can produce the theta burst stimulation on its own, or we can combine it with the nodal tDCS, that is the pink, or cathodal tDCS, that is the blue. What do we see? The black dots are without any tDCS. You can see that up to 20 minutes, there's a very stable field EPSP. What that means is that the connection between these 2 networks of neurons is stable. The EPSP, or this field EPSP size, is stable. It's stable until we get to 20 minutes. Then the theta burst stimulation is applied. You see that the black dots, it's a little bit overlaid, but I think you can make it out that the black dots go up. There's about a 60%, 70% change. And an hour later, though there's some decay, that connection remains stronger. So that is a basic experiment where theta burst stimulation produces plasticity, a change in connectivity between 2 populations that's durable. If we do this, if we apply the theta burst stimulation at the same time as direct current stimulation, we can get more bang for the buck. You can see that the pink dot is higher than the black dot. When we apply theta burst stimulation and we combine it with direct current stimulation, the theta burst stimulation produces more plasticity, more durable plasticity. And if we reverse it and apply the cathodal version of direct current stimulation, we can produce the opposite. It's a weaker effect, but we can produce some suppression of the plasticity. What's not shown here, which is really critical, is that if you apply the direct current stimulation on its own without theta burst, you don't produce plasticity. You can apply it now until the cows come home. Direct current stimulation in our hands on its own does not produce plasticity in the brain slice. Only synapses that are already undergoing plasticity have the possibility to benefit from it. And we've unpacked this in a lot more detail. We've looked, for example, so in this case, there's an experiment where you have 2 populations of neurons synapsing onto the same cell. One is quiet, the other one is active and undergoing plasticity. Now we apply direct current stimulation. On the same neuron, or the same population of neuron, only those synapses undergoing plasticity get the benefit of direct current stimulation. The ones which are quiet, this is on the same neuron, we do not produce plasticity. And we can still be more nuanced. I know I'm going through this quickly, but the reference is there. So by the way, and this is functional targeting, right? This is functional targeting on like a cellular axon specific level. Only synapses that are undergoing plasticity get the benefit of direct current stimulation, more bang for the buck. The quiet ones don't. But it turns out there's other properties that we can probe in a brain slice. These relate to Hebbian plasticity and associativity. In this experiment, we have one pathway that is coming in and it is strongly activated and it's undergoing plasticity. We have another pathway coming onto the neuron. It's active, but weakly. So it's active, but in a way that is not enough to produce plasticity on its own. When we apply direct current stimulation, we can boost plasticity in the strong synapse and create plasticity in the active but less active one. And again, I'll refer you to the paper for the full paper, but what the overall picture that it's forming is, we like to say tDCS boosts Heb, or you have Hebbian plasticity and tDCS boosts that mechanism of plasticity. And this directly ties into the notion of functional targeting, but it brings up other ideas of associativity, you could call it transfer of effects, and so on. This work is part of literally decades of work that all show the same thing. tDCS on its own, not much of an effect. tDCS on an active system, you get an effect. And so the prediction, as far as the translational prediction, like what you should do now in a clinic, becomes this, that tDCS should not be applied on its own. You shouldn't apply tDCS to someone just sitting there in the room staring at the ceiling and hope that they get better at whatever it is that you want them to get better at. Rather, tDCS should be combined with a training or a task, and that task should be what you want them to get better at. So if you're trying to make them a better reader, then they should be reading while you apply tDCS. So that's the prediction. Now, in a sense, this is a big, like, whatever moment, because that's exactly how tDCS has been done all along, right? Going back, over a decade. I mentioned Julius Frederson's work, but if you look at the body of work of using tDCS in rehabilitation, it's never applied alone. So this prediction is a bit of like a, I don't know, we want cart after the horse kind of idea. This is already how tDCS is done, but I don't think it's not for nothing. I think this body of literature using animal models really has given us a very, very precise substrate for how tDCS works in these applications and why applying tDCS in the context of training produces an effect. And again, there's a lot more papers on this topic. I want to talk about one more idea. It is something that I've been thinking a lot about, and it's not about neurons. It's about something else. It's about brain vasculature. It's about the endothelial cells that form the blood vessels that go through the brain. And myself and others have become very interested in the idea that transcranial direct current stimulation is a very powerful booster of brain blood flow, a very powerful booster of transport across the brain. I mentioned earlier on in the talk that tDCS doesn't produce an overt muscle twitch or anything, but it does produce tingling and it does produce a lot of reddening. It's very well known. You put tDCS over the skin, you get a lot of reddening. Well, the question is, could the brain be reddening also? Could tDCS be producing some sort of hemodynamic vascular response at the level of the brain? And it turns out that the answer is yes. In fact, people have known this for a very long time. But let me, before I get to that, say a few more. There's this general notion of a neurovascular unit, and the concept that there's a very tight coupling between blood flow and metabolic activity, blood flow and neuronal health, blood flow and learning. And so this is this neurovascular coupling, and it's a two-way interaction where, on the one hand, the blood supply is giving the neurons what they need, but on the other hand, the neurons are signaling, I need more blood supply, and so on. And I don't need to tell this group that there are many diseases that are associated with a dysfunction or an injury to the system. And so we have this neurovascular unit, and the question is, could tDCS be activating this neurovascular unit in some way? And again, in one sense, this question has been answered exhaustively because people have done tDCS in the context of things like fMRI or ASL, arterial spin labeling. And they've shown very conclusively that when you do tDCS, or even FNIRS, right? They've shown very conclusively that when you do tDCS, you get a change in the fMRI signal, which is a hemodynamic signal. However, these results have always been interpreted as tDCS modulated the neurons, which of course then drives a hemodynamic response. That's the whole principle behind fMRI. That's the classic order. Neurons first, blood supply follows. But the question we wanted to ask was, well, could it be the other way around? In this particular case, right, not normal, but in this exogenous case, could we apply stimulation where the blood supply comes first and then the neurons respond? So it's still neurovascular coupling, but in a different order. So I want to emphasize, there's no suggestion here that tDCS doesn't affect neurons. I just showed you data that tDCS affects neurons. In fact, brain slices have no blood supply. So it's a very clean example of showing how tDCS in the absence of blood supply can still have an effect. And as I mentioned, there's a lot of data showing indirectly that tDCS triggers changes in hemodynamic changes that you can image, but those are always considered epiphenomenon, right? tDCS produces a robust change in blood flow measured by ASL, but that's just because it activates the neurons. But the question we wanted to ask was, we'll take the neurons out of it. Just leave the blood supply. Could tDCS be directly activating blood flow? The way we addressed that question was by using a model, a cultured model of cells where there were no neurons. It was simply a blood-brain barrier. It was just endothelial cells. And we had to do the experiment this way because if you do any other experiment in the intact brain and you see both neurons respond and a vascular response, People will attribute the vascular response as an epiphenomenon. Neurovascular coupling, of course you see it. You've activated neurons, so the blood supply goes. So the only way we could prove that there was a direct vascular response was by taking the neurons out of the equation. And there are different model systems to do that. One of them is simply culturing endothelial cells in a dish. And so we have that model system available to us. The cells are in a dish, and we can actually put electrodes across that dish and directly zap this BBB, this blood-brain barrier model, to ask in the absence of neurons, do endothelial cells, does the blood-brain barrier respond? And there's a lot of markers we can use looking at transport of water, transport of different molecules, also looking to see if there's any kind of morphological changes or gene expression changes. And a lot of those details are in the paper, but to make a long story short, we saw very robust responses. More than that, we were able to explain those responses. So what you're seeing here now is water transport across the blood-brain barrier. And you can see that when there's the bubble displacement is like a bubble that's tracking flow. So as the bubble is moving, it means that there's a constant flow. And if there's a change in the rate that that bubble is moving, it means that there's a change in flow itself. So in panel A, you see the bubble movement. And in panel B, you just see the flow itself. There's that initial transient in the beginning in B. That's just because the endothelial cells are sealing over time in this model system. But by eye, very obvious, trial by trial basis, you can see that when you apply direct current stimulation, you change the flow. It's intensity dependent. It's actually linear with intensity. It's polarity dependent, meaning the anodal or cathodal variation will push water flow in the different region. And it can be precisely explained by theories of something called electroosmosis, where it's very well known that electrical current flowing through a tight barrier will drag water through it. It can be quantitatively explained by this theory. Moreover, as that water starts to go through, it drags things with it. It can drag different molecules with it. And depending on how that water is flowing, different molecules will be impacted differently. And it turns out that this rush of water flow also led to a whole range of gene activation. And so what this paper showed is that there are both acute and lasting effects of direct current stimulation directly on the walls of the blood vessels, the blood-brain barrier, in the absence of neurons. So this is really my last slide. I'm really excited about this idea as a new direction, a new translational direction for tDCS. There's a lot of work coming out. This is just another work from our lab. This is now in vivo, so using multiphoton imaging through the skull to image the blood-brain barrier, showing that when you apply direct current stimulation, you change you change flow, you change transport across the blood-brain barrier, and that's going to have a very profound effect on neuronal capacity for plasticity, neuronal excitability, and so on. There's many questions to unpack, but I think what is really becoming clear at this point is that there's a direct effect on vasculature, and we know vasculature has a huge effect on neuronal function. There's a interesting aspect of this that this can also drive brain clearance. So there's a lot of interest in brain clearance mechanisms. These are known to be impaired in aging. They're known to activate during sleep. And there's many neurodegenerative disorders that are linked to an inhibition in brain clearance mechanisms. And now we have a technique that can directly drive brain clearance. This electroosmotic push will drive water through the system, essentially flush the brain, clear things out with it. And we have several publications, as do others, addressing this point. And the final point is that this notion that tDCS can produce an immediate and profound effect on blood flow has suggested that it may be used in a very different context, in a neuroprotective context. This is a study, this is a reference to a review of animal models, but there are currently clinical trials running using tDCS in an emergency capacity within an hour of the injury, where the tDCS is being put on the damaged region and is trying to influence it in different ways, including by trying to draw more blood to a region that has been injured. So for example, you can have a perilesional region that is vulnerable, and now you're drawing blood to that region and potentially producing a protective role. So this is a family of independent but related effects, and I wanted to finish here because for me, this is a big watch this space because the data is coming out hot and fast, and I'm very excited about it as a new avenue. So I'll finish there. I wanted to thank you for your attention. And again, I wish I could see all of you in person, but in another day. And for those who are interested, I did put the slides online as well as links to the references. So thank you.